Welcome to this Convergence Science Network event. My name is Luana Smahil. I'm the Executive Director of the network. It's a great pleasure to see so many of you here this evening. Let me introduce our speaker. Joel Kramer is a neuropsychologist and Director of the Neuropsychology Program at the University of California, San Francisco Memory and Aging Center, where he helps diagnose and care for patients with a wide range of memory disorders. In his research, Dr. Kramer studies the cognitive and behavioural effects of neurodegenerative disease, behavioural markers of preclinical Alzheimer's disease, and cognitive changes associated with typical ageing. Please welcome Dr. Kramer.
Thank you so much for the introduction. Thank you so much for inviting me. It's an honor to be here. It's also great to be back in Melbourne. So I'm here basically to talk a little bit about cognitive aging and describe a little bit about my model and how I conceptualize cognitive aging. First, the acknowledgements. So most of the funding comes from the National Institute of Health. There's also some philanthropy. The Hillblum Foundation, we have an aging network grant that's been instrumental in funding a lot of the work that I'll be describing today, also funding from the Tau Consortium. And then finally, I have to disclose a small relationship with Pearson Incorporation, which involves some occasional royalties. All right, here are the real acknowledgements. So this is the Memory and Aging Center. This is the picture at our last retreat. We're now about 150 folks. It's a fairly multidisciplinary or transdisciplinary group. We have lots of behavioral neurologists and neuropsychologists and nurses. We have imaging experts and a huge information technology team and an army of research coordinators who are really largely responsible for all the work that I'm about to describe. Also a fairly robust and active training program for all disciplines, ranging, again, from neurology to pharmacy to nursing to neuropsychology. We are at for those of you who know UC San Francisco Med Center, there's a new campus that's very much neuroscience oriented. This is the neuroscience building where the Memory and Aging Center is located. And this is part of the aging team. Although from the looks of it, I'm really the only one who's actually aging. But they, again, are really, drive a lot of the work. All right, let's talk about cognitive decline. So this is based on real data. I think this is a memory test, but it doesn't really matter. It could be a reasoning task, a processing speed task, a visual spatial task. It really doesn't matter what the cognitive task is, with the exception of maybe language and wisdom. But you can see there is a very gradual, steady decline with age, what I oftentimes refer to as the slippery slope of senescence. And this is what we're taught, right? And again, this is real data, cross-sectional data ranging from 20 to 90. And how many of you are actually taught that this is what to expect with aging? Just raise your hand. All right. So probably the majority of you. I say it's bunk. I say it's completely wrong. Why do I say it's wrong? Why do I refer to this as the myth of cognitive decline? Well, this is a regression line. This is sort of a summed average of all the data. Here are the actual data points on which that line is based. Take a look at that 90-year-old who's just knocking this test out of the park and performing so much better than most of the 20-year-olds. So what does that really mean? This is, we're seeing a tremendous amount of heterogeneity or variability. Now this is cross-sectional data, right? So we haven't followed any individuals over time. So we should actually look at some of our longitudinal data. This is data from, I think, an executive task over a 2 to 3 year period. And this is a spaghetti plot of how people did over time. And when you look at it, it pretty much looks like spaghetti. But when you sort of tease apart some of the variability and the different kinds of slopes, what you can actually tease apart are some very different, distinct cognitive aging trajectories. So not everyone is aging at the same rate or at the same amount. Now we all want to be in that blue line, the red line. you know, stable over time, not changing at all. You know, these three lines probably represent the range of typical aging. Some people do a little bit less well than others, but everyone's pretty well functioning. And then you've got a few people who are actually just dropping fairly rapidly. Highly likely that these people have a neurodegenerative disease. But you got to remember, we're oftentimes seeing these people at the very early stage, and the challenge now, of course, is to predict which trajectory someone is about to go along. All right, so here are basically the take-home points of what I've talked about so far. So decline in cognition with age is not inevitable. There's a tremendous amount of variability in how much and when. These age-related changes in cognition and brain, so this brain structure, brain function, in my view, are largely pathological. Right? It may be age-related pathology, but it's still pathological. Our brains are no different from our hearts or our knees or our backs. There are age-related changes, but let's face it, when your knees hurt, you don't just say, oh, this is just aging. You want to figure out what are the pathological substrates of that and figure out what we can do to ameliorate those age-related changes, right? Because if we understand the biology of these changes, we can then start thinking about maybe we can do something about them, all right? So anyway, that's sort of the crux of my position here. All right, so are we with me so far? Okay. So why is this important? Well, the reason why it's important is because when you look around the world, the number of people living into their 70s and 80s and 90s is mushrooming. Now, these data are US data, but I don't think they're going to be much different from most countries. Certainly not Australia, certainly not Europe, certainly not China, it's not Japan, not Cuba. Basically, what we're seeing is a huge bolus, or a huge increase in the number of people living over the age of 65. And you know, another sort of huge increase in the number of people living over the age of 85, to the point where the number of people over the age 85 in the States is going to be well over 10 million in just the next few decades. And you also have to consider the fact that the incidence of dementia or significant cognitive impairment doubles every decade. So that by the time someone's in this 85 and over group, there's a 30 to 40% chance that they have significant cognitive impairment or a dementia, which means that this silver tsunami is leading into what's going to essentially bankrupt healthcare systems across the world unless we can do something about it. And anything that we can do to even delay the onset of cognitive decline will save billions of dollars in healthcare costs. So that's why it's critically important. So here's my vision, and I don't know if this will actually ever come to be, but here's what I imagine, that you're 70 years old, you go see your physician, it's your annual wellness visit. Every year for the last few years, you've been going in and having a cognitive screen, and your internist has ordered blood tests, and you filled out questionnaires on your lifestyle, and we've sent your DNA off for analysis, and you've had imaging, and your physicians can really sit down with you and say, look, based on what your brain scan looks like, and based on your DNA, and based on how you did on this last round of cognitive tests, and what your lifestyle and inflammatory markers, et cetera, are, I think that in the next 5 years, you are at increased risk for such and such, and here are 5 things you need to do about it. We already do this for cardiovascular health. So the next phase is to start doing this for brain health on a very large global scale. So this is a tall order. It's highly multidisciplinary or transdisciplinary effort. We're starting to try and pull together a large cohort at UCSF. So far, we're probably up to around 650 functionally intact community-dwelling older folks. I don't like to use the word normal aging because, as I've implied, I think a lot of aging is pathological. But we can talk about it as typical aging. And so we're following now close to about 400. We have DNA on everyone. We have cognitive screening. We have neuroimaging. We're starting to get amyloid imaging. This is looking for Alzheimer's proteins in the brain. We're starting to get more cerebrospinal fluid, which really tells us a lot about what's going on in the brain. And we're going to follow these folks until forever, until autopsy. All right, so how should we start thinking about aging? So let me propose a working model. So in this working model, basically the bottom half is brain structure and brain function, right? We have all these cool things that our brain does. We can use language and nonverbal skills, and we can do reasoning and multitasking, and we can remember things. And all these amazing cognitive things are mediated by brain structure. Our brain has gray matter and white matter. We have the hippocampus, which is really important for episodic memory. And this is where neuropsychologists typically dwell. But in this aging model, we start to go back a little bit further to think about, well, what might be some of the biological events that predispose people for some of those adverse aging trajectories? Things like cerebrovascular risk factors, inflammation, proteins, which I'll talk about in a little bit. And what are some of the things that influence how much of this there is? Well, we've got genetics, we've got lifestyle, and we have a lifetime of health and exposures. So I'd like to talk a little bit more in the next 45 minutes about some of the details of this model. Two caveats I need to mention about this model. One, it's horribly oversimplified. Right? I've left out tons of boxes, and I've left out lots of the relationships between those things. I haven't even broached the subject of interactions between these various boxes. And the other thing I have to acknowledge is, you can see lots of directional arrows on this graph. And again, that's also hubris, because it implies that there's unidirectional causality, and we know that's probably not true. It also implies that I actually know what those causal relationships are. And when I present data to you today, most of that data is based on either cross-sectional or very short-term longitudinal data, and much of it is observational data. It's not hardcore experimental data. So I can't say with confidence that X causes Y. In fact, some of the time, X may just be an irrelevant bystander, but I'll talk about some of the relationships between X and Y. All right, so let's start thinking about the model. And I'm just going to go through some of what I think are the more salient features of it, and leave time for some questions at the end. All right, genetics. So, I think the genetics of cognitive aging is just beginning. And we know so little now about some of the genetic protective and risk factors for how our brains age. But there are a few that we know more about. We can talk about ApoE. Now, the ApoE gene is the one gene that we know is strongly associated with risk for Alzheimer's disease. So, and this is some of the work by Rick Caselli and Eric Ryman at the Banner Institute, and I think that this is the slide to really look at, which really basically says if you have two of the E4 alleles of the ApoE gene, you're going to have one of those adverse cognitive aging trajectories. You're at much greater risk for cognitive decline, even more so than if you have just one of the E4 alleles, and then this is what the longitudinal data look like if you don't have any of those E4 alleles. You're much more likely to have stable cognition over time. So I think ApoE is a fairly big one and the one that we know most about. But there are lots of other genes that I think are really interesting and probably have a significant impact on how our brains age. So this is work done by two folks at the Memory and Aging Center. This is Dina Dubal on the left, who's a bench scientist and works with mouse models. This is Jen Yokoyama, who is a geneticist. And they looked at the Klotho gene. Now, the Klotho gene is well known in animals. So about 25% of us are heterozygotes for the Klotho gene. So you have two different kinds of amino acids. So one from your mom, one from your dad, they're different. And if you are a heterozygote, if you're one of those lucky 25%, you age better. So it's pretty well known in the mouse literature that mice live longer, they live more healthily, they have higher levels of the klotho protein in their blood. So one of the things that Dina and Jen were very interested in is to see what impact the klotho gene had on cognition and brain structure in our older population. So they combined data from a few different sources. And one of the things they described is that if you were a heterozygote for klotho, you had a larger dorsolateral prefrontal cortex on the right. You had bigger brain regions, right? Great, good start. What is the dorsolateral prefrontal cortex so important for? Well, it's really important for executive functioning, our ability to organize and plan and multitask. So the next thing they did is put together, based on the available data, some composite scores for executive functioning, and found that there again, the heterozygotes did better than those who didn't carry that particular gene pattern on measures of executive functioning. So the next question then really is, does this help with the cognitive aging trajectory, or is it, the people who are heterozygous just started off better and they're going to stay better? Well, we don't really know that yet because this is all cross-sectional data. But the cross-sectional data do kind of hint in one direction. So the purple line are the heterozygotes, and you can see that they're doing better across the age spectrum than those who are the non-carriers. Now, if this was going to help one's brain aging trajectory, you'd think that those two lines would get further and further apart with greater age. But in fact, they're getting closer and closer together, suggesting, again, with cross-sectional data, that maybe klotho is a gene that gives your brain a head start, but it doesn't really help you as you age. But all these subjects are now being followed longitudinally so we can have a more direct measure of the impact of the klotho gene on brain aging. Brain-derived neurotrophic factor is another, I think, really, really interesting gene. Now, brain-derived neurotrophic factor is something, it's a hormone, it's a growth factor that your body produces. It's well distributed in the brain, particularly the hippocampus, which I mentioned is very, very important for memory. And one of the things that brain-derived neurotrophic factor helps with is creating new neurons. I mean, I remember when I was first learning neuropsychology, I was taught that we never made any new neurons as we got older, but that's not true. We actually do have the capacity to generate new neurons. BDNF is something that's very important for that. And there's genetic variability in the BDNF gene, which influences how that BDNF protein looks like. So about 2/3 of us are homozygous for valine, and that's good. You want that. Now, the other option on this one particular SNP in the BDNF gene is methionine. So if you have one or two methionine amino acids, that's less good. All right? So we wanted to look at the difference between people who are homozygotic for valine versus those who had one or two of the methionines. Now, we know from the literature that people who have a methionine are at greater risk for psychiatric problems, but we were particularly interested in cognition. And given the role of brain-derived neurotrophic factor in the hippocampus, given the hippocampus's role in episodic memory, we were interested to see if there were any group differences in how well these people were able to hold on to information over delay, because that's what the hippocampus does. It enables you to hold on to information over time. So we gave them a list learning task. And again, I have to remind you, these are all functionally intact folks, and they all performed pretty much in the average range. So we're not talking about deficits here. But when you compare the two groups on how they did during the learning trials, there were absolutely no differences whatsoever. So this is the valine group in red, this is the methionine group in blue, and over multiple trials of a list learning task, they are performing pretty much at the same. Now, what happened after a 20-minute delay? Well, what we found is that if you had one or two of the methionine alleles, you are less able to hold on to information over delay, consistent with the notion that BDNF plays a very important role in hippocampal functioning. So again, an example of how normal genetic variability impacts cognition over time. And again, all these folks will be followed longitudinally to see if this predicts these brain aging trajectories. There are multiple other genes that I think are particularly interested and relevant for brain functioning. Dopamine genes, serotonergic genes, and all of these are things that we're going to be following over time. All right, enough of genes. Let's get back to the model and now look a little bit at proteins. All right, so why are proteins so important? Well, first of all, sometimes proteins start to misfold, they start to clump together, and these misfolded aggregated proteins are really what causes the neurodegenerative diseases that are so common as we get older. So the two proteins that are really important for Alzheimer's disease are amyloid and tau, but there are a whole host of other proteins that, when misfolded and aggregate, are associated with neurodegenerative diseases. Protein called TDP43 is associated with Lou Gehrig's disease, or ALS, also some of the progressive aphasias, behavioral variant frontotemporal dementia. There's specific species of tau that are associated with some other neurodegenerative diseases. Alpha-synuclein is the protein that causes Parkinson's disease. So if I'm talking about typical aging, why am I talking about neurodegenerative diseases? Well, let me explain. We are now able to use molecular neuroimaging to visualize the amount of amyloid plaque in someone's brain. All right, so here's a typical patient of Alzheimer's disease, and you can see in red essentially the regions of the brain where there are these abnormal accumulations of amyloid plaque. All right, pretty symptomatic. You wouldn't want your brain to look like that. And then on top, here's an age-matched normal control who doesn't have any amyloid. Great. How about these other folks? This is a cognitively normal individual whose brain is riddled with amyloid. So does this have any implications for how this particular person is going to age over time? Well, the answer is probably. Well, first of all, the number of normal or typical aged individuals who have amyloid in their brain really increases a lot with age. So that by the time you're in your 80s, you have about a 40% chance of having a lot of amyloid in your brain, and a scan that looks like someone with Alzheimer's disease. Now, is this amyloid accumulation fairly benign or innocuous? And the answer is probably not. So here's what we know about having amyloid in your brain, that it is a risk factor for adverse cognitive decline over time. So when you start to follow folks who have these positive amyloid scans. Right? Now, the other protein that's particularly relevant for Alzheimer's disease, as well as a whole host of other syndromes, is tau. And the capacity to image tau has just been developed over the last couple of years. We are now starting to do tau imaging of some of our normal controls, and we can start seeing some tau aggregates. And those tau aggregates happen to be in those regions of the brain where tau first starts to accumulate in Alzheimer's disease, around the hippocampi. So you can see some of the tau aggregates around the hippocampi. So again, is this tau benign? Probably not. In fact, some of the early work suggests that the more tau you have, the less well you're going to do on memory tests. So I think that when you have a population of intact, community-dwelling elderly, again, if they're over the age of 80, probably 30 to 40% have accumulations of these proteins in their brain that is most likely affecting how their brains are going to age over the next few years. There are other proteins in the brain that are not misfolding or aggregating that probably have an impact on cognition. So this is testosterone over the lifespan in a typical male. So it really starts to sink until it's, by the time we're in our 80s, it's It's less than 1/2 of what it was when we were in our 20s. A lot of downsides of that low testosterone, but we also know that testosterone levels are associated with how well men do on visual spatial tasks. And here's sort of an interesting trial of older folks who were given either placebo or testosterone, and this is their performance on visual spatial tasks. And this is one of those where the lower you are on the bar, the better you're doing. And as you can see, the treated group started doing better and better over a 6-week period than those who were on placebo. So again, typical sex hormone is directly associated with cognition and probably influencing how one does over time cognitively. Estrogen. Estrogen is a pretty powerful protein. I had the honor of watching what a big bolus of estrogen did to my daughters. But we also know that estrogen has significant impact on the brain. We know from human models as well as mouse models that it's neuroprotective, particularly in the hippocampus. The higher levels of estrogen are associated with sort of healthier neurons, that premenopausal women and mice with estrogen on board do better following a stroke or some kind of brain ischemic event. And there's some interesting studies with women in their menstrual cycle suggesting that they do better on several cognitive tasks during the phase of the menstrual cycle where estrogen is at its highest levels. So we were kind of interested in the effect of being pre or post-menopausal on a verbal memory task. So we basically looked at a delayed recall task. And again, this is cross-sectional data. So again, this is one of those times where I can describe a correlation, but not necessarily imply causality, though I probably will imply causality. So one of the first things we did is we basically said, well, how well are the men and women doing on this memory task? And this recapitulated lots of results that showed that women do better on verbal memory tasks than men, right? The next thing we did was said, all right, well, let's identify the period during which most women on average go through menopause and take that out of the equation. So now we have 4 different regression lines, and we wanted to see what the relationships were between age and memory in men and women pre and post menopausal phase. So if you look at men prior to this particular, the younger men prior to age 50, there's definitely a negative relationship with age and memory. The older the men got, the less well they did. But in the women, there was absolutely no relationship with age whatsoever. And you can actually just see that from the regression line. It's pretty flat going across up until age 50. After age 50, men continue to show decline, and women also have comparable rates of memory decline as a function of age. Consistent with a lot of observations that talk about the impact of say, hormone replacement therapy on maintaining optimal memory performance. Now, hormone replacement therapy is an extraordinarily complicated and controversial area, so this is certainly not a recommendation for hormone replacement therapy, but just an illustration of how a circulating sex hormone can potentially have an effect on cognition, and age-related changes in hormone levels can be predictive of age-related changes in cognition. Health and exposures. So health and exposures for me sums up all the stuff that happens to our brains as we get older. And this is stuff that happens early on. Now, there's some positive things. I mean, I think that lots of education when you're young is probably a good thing for your brain and associated with a better age-related cognitive trajectory. And we're only beginning to scratch the surface to understand what some of these early exposures might be and what the long-term impact is. All those times in adolescence where you drank too much or smoked a lot of dope, are these really benign events, or could they potentially have some long-term consequences on brain structure and function when we're in our 60s or 70s? Well, I do want to talk about a couple of areas that we do have some more data on. One is the impact of having a cancer diagnosis. So this is some work done by Laura Heflin, who was a postdoc in our group, who looked at a Swedish twin registry and had a fairly large number of twins over the age of 65. And she looked for those who were discordant for cancer. So again, some of the twins had cancer, some did not. And she was interested to see what impact this exposure to cancer earlier on in life had on the risk of cognitive decline or dementia, again, many, many, many years later. And in fact showed that when they were given a cognitive screen, the twin with the cancer history did less well than the twin without the cancer history, again, starting in the 60s all the way up until the 80s. So, what are the biological mechanisms by which this cancer history affected brain structure and function? Not all that clear, but I think it sort of raises the question of early life events and what impact that might have. Now, another topic that's been of big interest is traumatic brain injury. So I know that Aussie-style football is associated with lots of injuries. We have the National Football League in the States, and this whole idea of chronic traumatic encephalopathy is in the news all the time. Is an early traumatic brain injury meaningful? Does it have long-term consequences on how your brain is going to evolve in your 60s, 70s, and 80s? So David Perry, one of our behavioral neurologists, just spent months combing through the literature to do a meta-analysis on whether a mild traumatic brain injury, and most of these studies were about a single mild traumatic brain injury, and whether this was associated with a greater risk for a neuropsychiatric sort of illness later on in life, and basically found that although the odds ratios were small, they were consistently significant. So there was a higher odds ratio for dementia in general, Alzheimer's disease, Parkinson's disease, ALS, as well as depression, and just general psychiatric conditions. Again, he emphasizes that the odds ratios were still small. The odds ratios for the traumatic brain injury were, say, less than pesticide exposure for Parkinson's disease, but still significant, and supporting the idea that even early hits to the head may have adverse consequences 50, 60 years later. We know that the Boston University group, Bob Stern, recently had a paper out suggesting that kids who were playing football early on were at a greater risk for having chronic traumatic encephalopathy later. So maybe there is something about these exposures to a developing brain that may not rear its head until 50, 60 years later. There's another study done by Raquel Gardner, another behavioral neurologist in our group, who looked big. She took a very big data set to quantify whether or not a traumatic brain injury later in life or was associated with increased risk of dementia later on. So she looked at a California statewide registry where there were just tens of thousands of individuals who had exposure to a traumatic event, and she looked for the years 2005, 2006, had over 150,000 individuals who were, had some kind of traumatic event, and then characterized them as having either a mild or moderate traumatic brain injury or a non-head injury. This was usually sort of a fracture of some sort. Then looked to see if there was a dementia diagnosis 3 to 4 years down the road. All right, so here's what she found. She had over 50,000 people who had a traumatic brain injury, over 100,000 who had some peripheral fracture. TBI group maybe was a little older, had a little more alcohol, more males. But when you actually looked at the nature of their injury, it was mostly falls and then some motor vehicle accidents. But the group that had the brain injury had an 8.4% rate of dementia diagnosis, as opposed to only 5.9% for those who had peripheral fractures. So again, highly significant. And for those who did go on to have a dementia, the time between the injury and the dementia diagnosis was shorter for the group that had the mild traumatic brain injury. So again, there's a good strong suggestion that even outside the realm of the football arena, a traumatic brain injury is associated with some increased risk for one of those negative brain aging trajectories. All right, that's early life exposures. How about cardiovascular risk factors? Well, we all know what the cardiovascular risk factors are, and certainly having cerebrovascular disease can be associated with some cognitive problems. So the two typical things we look for when we're measuring cerebrovascular disease are these tiny little infarcts, tiny little strokes that are outlined here, or changes to the white matter that are usually ischemic in nature. These were for a while called silent strokes because they were asymptomatic. And when you take a group of 75-year-olds who deny any history of stroke and you do MRI scans on all of them, you will find these, evidence for these tiny strokes in about 20 to 25%. And you'll see lots of white matter disease, and again, in about 20-25%. So it's not uncommon in a typical aging cohort. Are these strokes silent? Absolutely not. There's plenty of data out there, including some from our group, that really suggests that these silent strokes are associated with some cognitive problems, even if all of these individuals are functioning adequately in the community. We were interested in some cardiovascular risk factors that weren't necessarily associated with measurable cerebrovascular disease on an MRI scan. So this was a study done by Darvis Fraser in our group, who actually wanted to look at insulin resistance. Right? So we took a group of, around 150 individuals. He looked at some measures of executive functioning, again, working memory and problem solving, organization, and wanted to see if a measure of insulin resistance was associated with cognition. So he took a fairly conservative approach. He said, well, all right, let's control for everything in our model. So he controlled for age, he controlled for education levels, he controlled for gender, he controlled for other cardiovascular risk factors like cholesterol, he controlled for body mass index. This is mean arterial pressure, so how much blood pressure the person had. He actually went in and said, all right, let's control for the amount of white matter signal hyperintensity there was in the brain. So he pretty much controlled for everything you can think of, and he still found that there was a significant relationship between the homeostatic model marker of insulin resistance and executive functioning. So again, how is it that insulin resistance is affecting brain structure and brain function? Not really clear, but something that we are definitely pursuing. And then actually we had a med student who came by who just worked for us for the summer and said he was kind of interested in this topic. So he looked at triglycerides and basically found the same thing, that someone's level of lipids in plasma was associated with poorer performance on executive tasks, even when everything else was controlled for. And then one more study, again done by Darvis Fraser, that got published last year. We were interested in the carotid medial wall or intimal wall thickness, right? So again, the thicker the arterial vessels are in your carotid arteries, it's a good marker of atherosclerosis. It's definitely a vascular risk factor. We looked at a large number of normals and used ultrasound to measure the carotid intimal wall thickness. And this time we were interested, we used longitudinal data. So the question was now not whether atherosclerosis and a cardiovascular risk factor was associated with poorer cognition. He asked, is it associated with rates of decline in cognition? And that's exactly what he found, so that the more evidence for atherosclerosis there was, the greater the decline in cognition over a 2 to 3 year period. It's cardiovascular. So let me spend a little bit of time talking about inflammation. So I think inflammation is going to be one of the more interesting topics. So inflammation is a normal response to injury, right? But the older we get, the longer or more sustained this inflammation is. And it doesn't matter whether it's a peripheral injury or what have you. We now know that inflammation peripherally is associated with an uptick in inflammation in your brain. So is this something benign? Well, I went to a talk once a few years ago where someone was describing their mouse model showing that higher levels of inflammation in his older mice was associated with poor hippocampal functioning. And we thought, we got to go home and do something and take a look at this. So we had measures of C-reactive protein, an inflammatory marker, in blood. That was not the most sensitive measure of C-reactive protein. So we basically had two groups. One group that we weren't able to detect any C-reactive protein, and a second group where we were. So the question now was, are we also able to see some evidence for changes in brain structure and functioning as a result of having measurable C-reactive protein? And the answer was yes. When you looked at memory performance, the group that had higher levels of C-reactive protein were less able to hold on to information on a memory test over delays, consistent with that mouse work that talked about some changes in hippocampal functioning. And when we actually measured the medial temporal lobes, we found that the group that had higher levels of C-reactive protein had smaller medial temporal lobes. Boettcher has gone on to show that higher levels of inflammation, in this case IL-6, is associated with changes to the white matter in the brain. Melanie Stevens has shown, again, looking at our longitudinal data, that again, in our normals, those who have higher levels of IL-6 at baseline had a worse aging trajectory on a measure of processing speed over a 2 to 3 year period. And eotaxin, another one of those fairly interesting inflammatory chemokines that we can now measure. So a couple of interesting things about eotaxin. Now, one is that our levels of eotaxin go up with age. This is cross-sectional data in humans. And you can do a lot with eotaxin in mouse models. So this is work done by Saul Viega and Tony Weiss-Correa, where they were looking to see what happens when you take young blood and put it into an old mouse. And they did this in a variety of ways. They actually tied the vascular supply of a young mouse and an old mouse together. And then they would also just inject into an old mouse, they would inject young mouse blood or old mouse blood just to see what would happen. Well, what would happen is nicely illustrated on this particular slide here. This is a memory test for mice, and this is marking the number of errors on how well a mouse was able to figure out where a hidden platform was. And you can see that when they took an old mouse and injected young plasma, that these older mice did better on the memory test than when they got another bolus of old plasma, right? Which has, and again in humans, we also see the same association between higher levels of eotaxin, worse performance on memory test. So the treatment implications are major here. So just put some young blood into an older animal. All right, that's, let me just briefly breeze through lifestyle because I want to be able to save time for some questions. Lifestyle is important to think about because we can do something about our lifestyle. It has an impact. One of the things that colleague Christine Yaffe showed several years ago is that when you follow folks who were measured, whose body mass index was measured in midlife, and followed them for 30, 40 years, what you find is that the group that had the higher body mass index had a much higher incidence of dementia later on. And in fact, it's not just body mass index, because they showed in a later study that it's the amount of abdominal fat that these individuals had that was a clear identified risk factor for dementia later on in life. Certainly body mass index in our hands is associated with more white matter disease. Another important lifestyle variable that clearly impacts cognitive aging is exercise. There are lots of epidemiological studies that suggest that the older folks who exercise more have a lower incidence of cognitive decline or dementia later on in life. And there's some very nice mouse studies from Karl Kottmann that illustrate this quite nicely. So he had two rats, one that got to run a lot, one that just sat around and watched I Love Lucy reruns. And he then sacrificed them and looked at the hippocampus. And one of the things that he had predicted was that in the hippocampus of the mice that exercises, you could see in red more staining for brain-derived neurotrophic factor, because one of the things we know is that if you want to up your levels of brain-derived neurotrophic factor, you exercise. Right? So not only did these mice have more BDNF in the hippocampus, but they function better. So not only did the young mice who were programmed to have Alzheimer's disease later on do better because they exercised, but if you took an older mouse and got them to start exercising later on, they too did better on cognitive measures than mice that did not exercise. This has been translated to humans. Art Kramer, who's in Illinois, did a randomized study with older adults. There was a stretching condition and an aerobic condition, and those who were in the aerobic condition not only did better on the memory testing, but also had healthier looking hippocampi on imaging and had higher levels of brain-derived neurotrophic factor in their serum. So I think that these lifestyle variables have a huge impact on how our brains age. So one last thing, this is a study done by Nick Bott, a student in our group, who was very interested in superagers, folks who just seem to they started off well and they continue to do well. So for us, information processing speed is one of those cognitive skills that is most sensitive to age. So he looked at our information processing speed data and basically identified three groups. So this is information processing speed. So what you want is to be fast, and by fast I mean you're lower down in the bar. So this is the superager group who are really performing on these processing speed tasks kind of like our 20 and 30-year-olds. And then there's a couple of groups who at baseline were performing average for their age. And then he followed these groups over a 2 to 3 year period. The superagers started off well and they stayed well. The average agers started off average and stayed average. And then there was a group that started off average and was appreciably slower 2.5 years later. So his basic question is, well, what's different about those three groups and what can that teach us? And how could we use that information? So he looked at their genes. I thought that they were going to have less ApoE4, completely wrong. There are no differences in cholesterol or some other cardiovascular risk factors. What he found is, well, first of all, they had healthier looking brains. This is a marker of the microscopic integrity of the white matter in their brains. And the superagers just had healthier looking white matter for whatever reason. Then he looked at their fitness. And the superagers were just more fit. They exercised more than the other two groups. And then he looked at inflammation. He used IL-6 and found that the superagers at baseline had lower levels of this inflammatory molecule, followed by the average agers, and the worst agers had the highest levels of inflammation. Again, causality, I don't know. Maybe the poor agers had some disease that was causing inflammation, or perhaps the inflammation was predisposing them to some other kind of disease. The jury's out on that, but again, it's a nice marker for how studying really successful agers is giving us lots of clues about some of the things that predict those aging trajectories. So, to sum it all up, again, it's so important for us to understand who's going to be on what aging trajectory so that we could do something about it. We know that proteins are important, inflammation's important, oxidative stress, which we didn't really talk about, cerebrovascular disease is major. We know that all of these things are probably influenced by genetics, influenced by lifestyle, influenced by health histories and exposures while we're younger. So how can we pull this all together to do something about it? Well, it's clearly, it has to be a very multidisciplinary, we need advanced biostatistics. We need software engineers to help us deal with big data. We need people who work with mouse models and fly models and worm models. We need cognitive neuroscience. We need people who are experts in genetics. We need physicists, because it's the physicists who are really providing us with those ligands that we can use in PET scans that's allowing us to measure those proteins in the brain. So that's really key. Epidemiology, bioinformatics, molecular biology, it all comes together. But even that's not enough, because my particular bias is that we need all these disciplines, but the important take home is that we need to be able to do this in a certain way. First of all, we need to be able to follow cognition and all of these biological predictors on an ongoing basis. We can't just bring subjects into the lab once a year or once every other year. We need to be able to measure things like exercise and cognition and blood pressure and sleep, which I didn't talk about, on a continuous basis, which is why I think these advances in biometrics and biotechnology are going to be key players here in how we start to unravel the mysteries of cognitive aging. And I also think that all of these efforts have to come together to influence public health policy at a global level, because it's only by thinking about influencing public health policy that we can actually make a dent in this silver tsunami of aging and adverse brain health that's going to be so devastating over the next 50 years. And it's going to be small things. I mean, I think these helmet laws were passed years ago to reduce the number of acute brain injuries, but we now know that one of the payoffs of helmet laws is going to be probably a lower incidence of neurodegenerative disease 50 years later. So public health policies that reduce smoking, that improve cardiovascular health, that promote exercise, that promote social networking, that promotes cognitive activity. All these things I think are going to be really, really important for maintaining optimal brain health as we get older, and it's important at a global level. So thank you for your attention.
Another round of applause for Joel. Thank you.
Thank you.
In closing, I'd like to thank the network sponsors. Their support makes these events possible. So without their support, we wouldn't be here. So please join with me in thanking our sponsors and acknowledging their incredible generosity. Thank you. Thank you.